Hi everybody, this is a big picture view of muscle contraction because I know it's hard to keep that big picture in mind when you're also thinking about all the different components of it. So when you move your skeletal muscles, the ones that are attached to your skeleton and move your body around, including those that control speech, then there's a neuron in your spinal cord. The cell body is in your spinal cord. And it is told by the brain to send an action potential down its axon to the muscle. And as you can see in this picture, typically these axons, not always, but typically these axons divide and have many axon terminals. So even though there's only one signal that's being sent down from the motor neuron in the spinal cord, it is received by several different muscle cells, muscle fibers. This is different than the autonomic nervous system, in which you would have two neurons between the spinal cord and the target tissue. With skeletal muscle, there's only one neuron in that chain. Furthermore, the signal, in contrast to the autonomic nervous system, is always excitatory. That is, it's impossible for the nervous system to tell a muscle to stop contracting. All it can do is stop telling it to contract, which is very effective. It does make it not contract. So an action potential has traveled. As you know, it doesn't really travel, but is regenerated down this axon. And then when it gets to the axon terminal, you have really what happens at any axon terminal, right, is here you have the vesicles full of neurotransmitter, in this case acetylcholine, and then when you get to the axon terminal, voltage-gated calcium channels will open, and calcium will enter the cell and cause exocytosis of the neurotransmitter, as you learned before. So these little red dots are supposed to be acetylcholine that's now been released into the synapse, into the synaptic cleft, and it binds to its receptors, which on skeletal muscle are the nicotinic type. And this is a type that's an ionotropic receptor, which means that the receptor itself is also an ion channel. So acetylcholine binds to its receptor, and when two molecules of acetylcholine are bound, the channel opens, and both sodium and potassium can actually pass through this channel, but it doesn't really matter in real life about the potassium because sodium has a much greater driving force. And the movement of ions through that open channel during the brief time that it's open is dominated by the movement of sodium, and that's the only one shown in this picture. So sodium comes into the muscle cell and depolarizes it, and it has an action potential. And that action potential spreads along the muscle membrane, and muscles have these pockets in them called T-tubules to allow the depolarization to spread to the interior of the muscle. As that depolarization spreads, it activates these voltage-gated proteins, which are very much like voltage-gated ion channels. But here, the voltage-gated protein is on the T-tubule membrane, so on the muscle cell membrane, but it's physically connected to this other molecule, which with a very confusing name called the ryanodine receptor. But you can just think of it as a calcium channel. All right, so the ryanodine receptor, as soon as you get the voltage activating this dihydropyridine receptor, DHP is fine, then it activates the RYR and calcium flows out into the cell. So all you need to really think about here is that the depolarization, the spread of the action potential, has opened calcium channels. Okay? So the calcium, which is not coming from outside the cell, but rather from these sort of sacs of calcium within the cell called sarcoplasmic reticula. The calcium comes out into the cell, and then, of course, it can bind to troponin, and this is now the part of the process that you may be more familiar with from cell bio, that you get an increase in calcium in the cytoplasm, that it binds here to troponin, which they're abbreviating TN, and when troponin changes shape when bound by calcium, you then get a change of shape in tropomyosin, and that exposes, as it's always stated, a place on actin for myosin to bind. Once myosin is able to bind there, it will. So it always is sitting there waiting to bind, and it will bind there as soon as that site is activated. So once you have myosin binding to actin, then we're here at the contraction cycle, which has many names as well. And the contraction cycle, again, is something that I think we're familiar with in cell bio, but probably need to re-familiarize with. But basically, it's a cycle of myosin and actin attaching and then moving relative to each other and then reattaching. So if you imagine that this is the part of the myosin that's attaching to the actin, right? The sort of head, myosin head. So it binds to the actin, and then you have a power stroke, right? And then it binds to the actin again, power stroke. So you can see that myosin and actin are moving relative to each other, right? We're moving my hand relatively across the screen, right? So at the moment of letting go, you would expect the actin to just snap back to its original position, and actually that's exactly what will happen. And the reason that it doesn't happen is that even though in textbooks we always see one myosin head moving, actually there are hundreds or thousands of them, and at any given point, some of them are attached and some of them are released. So when one of them releases to rebind, there are others that are attached and holding it in place. So it's kind of like if you were doing tug of war and you let go of the rope to get a better grip, your team wouldn't immediately lose unless you were about to do so anyway. So there's this asynchronous binding and rebinding of the actin and myosin. And all this will continue, if we go back to the first slide here, all this will continue while we're getting action potentials in these motor neurons. And when the action potentials stop, then the depolarization, right, there's no acetylcholine release, there's no depolarization, and the calcium is being constantly pumped into the sarcoplasmic reticulum. So as soon as the action potentials stop, the calcium will still being pumped back in, but it'll be no longer being released. And so pretty soon there won't be enough calcium left in the cytoplasm of the cell to activate that whole cycle, and contraction stops. Okay, if you have any questions, let me know.